Good morning class. Today I will take your revision class of chapter 6 and 7. Let's start with the chapter 6 and it is seed, its structure and germination. Now we all know that seeds are made up of cotyledons and the outer layer of seed is called seed coat, which is made up of double layered structure, and outer layer is called testa and inner whitish layer is called tegmen. And with these two layers, inside with these two layer cotyledons protected, and the small scar is present on the upper side of the seed, and it is to which seed attached to the ovule, and it is called hilum. And this hilum is near this hilum, small microscopic pore is present, that is called micropyle, and this is very essential for the exchange of gases in seed at the time of germination, and it is also for the absorption of water for the process of germination. Now we all know that on the basis of presence of cotyledons, seeds are divided into two parts: dicotyledonous seed and monocotyledonous seed. A seed having two cotyledons, suppose this is, it is two cotyledons. This is, that type of seed is called dicotyledonous seed. And if seed is having single cotyledon, then that type of seed is called monocotyledonous seed. And with these dicotyledon and monocotyledon, one more layer is also present or absent in the seed, and it is the endosperm. If the endosperm is present in the seed, then that seed is called endospermic seed or albuminous seed. And if it is present, if it is absent in the seed, then that seed is called ex-albuminous or non-endospermic seed. This is the structure of dicotyledonous ex-albuminous or non-endospermic seed. The upper, this portion, this is the cotyledon. This portion is called cotyledons, and where the part where the cotyledons attach, that part is called embryonic part. And at the embryonic part, some parts are present. The upper part is called plumule, and it is the future stem. It is also called future stem because when it grows, it gives rise to the shoot system of the plant. And below the plumule, epicotyl is present. And in the same way, below the epicotyl, at the base of the cotyledon, hypocotyl is present, and after hypocotyl, radicle is present. Radicle is called future root because when it grows, it gives rise to the root system. So this is the structure of non-endospermic dicotyledonous seed, and example is the bean seed, and it is the maize seed. Maize seed is the monocotyledonous endospermic seed. This layer is called aileron layer, and this aileron layer is a proteinaceous in nature, and it divides the seed into two parts. This part is called cotyledonous part, and this part is called embryonic part. And in this embryonic part, plumule and radicle is present, and in this endospermic monocotyledon seed, this plumule and radicles are covered by a sheath. The plumule is covered by a sheath called coleoptile, whereas the radicle covered by the sheath called coleorhiza. This is all about the structure of monocotyledonous and dicotyledonous seed, and now the conditions necessary for the seed germination. The water, oxygen and proper temperature. These three conditions are necessary for the seed germination. When the seed absorbs water, hydrolysis of the food takes place inside the seed, and so it becomes, the cotyledons becomes active, and so that germination start in the seed. And when at the same time when they did not get, if they don't get the air or oxygen, the germination never takes place. So the oxygen is also very necessary, and with the oxygen proper temperature is also necessary for the seed germination. Without these three conditions, no germination takes place in the seed. And there are two types of germination in seed. It is epigeel germination and hypogeel germination. In epigeel germination, seed lifted above the ground because of the elongation of hypocotyl. Seed lifted above the ground because of the elongation of hypocotyl in epigeel germination, and this germination takes place in bean seed. While in maize seed, hypogeel germination takes place. In this germination, in hypogeel germination, no seed lifted above, it remains under the ground, and elongation of epicotyl takes place. In hypogeel germination, epicotyl elongates and seed remains under the ground. No, it does not lifted above the ground. This is all about the types of germination, epigeel germination, hypogeel germination, and this is all about the this chapter. You have to study the topics that I have explained you again in the revision class. So let's start with the chapter 7, revision of chapter 7. Revision of chapter 7, and it is respiration in plants. What is respiration? Respiration is the breakdown of the, it is a biochemical process which takes place in each and every cell inside our body, and it is in this process food completely break down in carbon dioxide and completely break down in presence of oxygen. If it is not completely break down, so if it is not completely break down, so that respiration takes place in absence of oxygen. And in both these reactions, energy released, whether in presence of oxygen or in absence of oxygen, but the strength of energy is more or less. So this is the respiration, means it is the completely breakdown of the food which releases energy and carbon dioxide. So the difference between respiration and breathing is that respiration is a biochemical process and breathing is the physical process. It is just exhaling and inhaling of air. It is a physical process. No any reactions takes place in breathing process. But because of breathing inside our body, biochemical reactions takes place, means breaking down of food takes place, and that breaking down of food is called respiration process. Now, on the basis of presence or absence of oxygen, respiration divides into two parts: aerobic respiration and anaerobic respiration. In aerobic respiration, this is a food. It completely breaks down into the carbon dioxide molecule, some molecules of water and energy. In this reaction, in this type of respiration, lots of energy released. While in anaerobic respiration, see here Ch-6, H12O6, it is a food and no oxygen is present here, and because of absence of oxygen, this food is not completely breaking down. C2, it breaks down into the alcohol and carbon dioxide. Sometimes it also breaking down in acid, lactic acid and carbon dioxide, and very few amount of energy released in anaerobic respiration. And the energy which released, it is in the form of ATP. ATP is the adenosine triphosphate, means to the adenine 3 molecules of phosphate attached, and this is the very active form of the ATP, and this phosphate is very efficient for the glucose molecule. When it attaches with the glucose molecule, it again becomes the adenosine diphosphate, and it is the inactive form of the adenosine. It is an inactive form of the energy. It is energy currency. It is also called energy currency adenosine triphosphate. And the energy which is released, it is in the form of ATP, adenosine triphosphate. Now, the fermentation process is also the example of anaerobic respiration. In fermentation process, anaerobic bacteria or yeast, means fungi, ferment and produces the lactic acid or alcohol in absence of oxygen. It is a type of anaerobic respiration. Now, the site of these respiration, where this respiration takes place in the cell. Suppose this is the cell and this is the cytoplasm and mitochondria. We all know that mitochondria is a powerhouse of the cell. Why it is powerhouse of the cell? Because it releases energy. Then how it releases energy? Aerobic respiration first of all takes place in cytoplasm. Respiration has two steps: glycolysis and Krebs cycle. Glycolysis is common in both the respiration, whether it is aerobic or anaerobic, and this glycolysis takes place in the cytoplasm of the cell. And after glycolysis, in aerobic respiration, the next step of the respiration takes place in the mitochondria, and it is Krebs cycle. And finally, the food completely breaks in and releases carbon dioxide, water, and 32 ATP of energy. Anaerobic respiration stops here. Only glycolysis takes place in anaerobic respiration. No Krebs cycle takes place in anaerobic respiration. So, site of anaerobic respiration is only the cytoplasm, and only one step takes place, and it is glycolysis. And site of aerobic respiration is cytoplasm and mitochondria. And two steps involved in aerobic respiration, it is glycolysis and Krebs cycle. Glycolysis takes place in cytoplasm, whereas the Krebs cycle takes place in the mitochondria. This is all about the respiration process of respiration in plants. There are some plants also which are mangrove, and in this type of plants, elongation of special type of roots coming out, and these roots are called pneumatophores. These pneumatophores are having small stomatic pores, and which are helpful in the process of respiration, means exchange of gases. This is all about this chapter. In this way I have completed the revision of chapter 6 and 7, and we will start with the new chapter in next class. Till then, thank you.